Good morning, everyone, and I'm happy to start our annual Schultz Endowed Lectures. So this year, we're very happy to have Jennifer Lippincott-Schwartz. I'm going to do more formal introduction, but before we go into it, I'm going to give the microphone to Vice Chancellor of Research, Rodolfo.
Thank you very much. Good morning, everyone. Thank you for coming to this great event. I'm very happy to be here and have a chance to welcome all of you. So, as we often do in this event, let me start by respectfully acknowledging and recognizing our responsibility to the original and current caretakers of this land, water, and air, the Cahuilla, Tomba, Luceño, and Serrano people, and all of their ancestors and descendants, past, present, and future. Today, this meeting place is home to many indigenous people from around the world, including members of our UCR faculty, students, and staff. We're truly grateful for the opportunity to live and work on these homelands. And for some of you that may be coming from far away, what these homelands are is what we sometimes are referred to as the Inland Empire. We comprises the San Bernardino and Riverside County. It's a huge area with a 5 million people population, which is one of the most diverse and fastest growing, not only in the state, but in the nation. Our regional economy, to give you an idea, is larger than that of, depending where you look, maybe 15 or 20 states of the Union. So people usually say that California, who has the fourth economy in the world, will be, as a nation, the fourth economic power. Well, within that nation of California, this will be a very strong state, the Inland Empire. As the only one university in the region and a proud member since 2 years ago of the Association of American Universities, our campus plays a very special role in the region. And we're leading the way in groundbreaking research and driving economic development to overcome many of the challenges we are facing in the region and our community, but which are not unique to this region. Our faculty, student, and partners develop solutions that address some of the most complex challenges faced in our world, from human health and sustainability to energy, agriculture, technology, and beyond, creating meaningful impact not only locally, but in a way that can be scaled at the national and global level. Some of our recent accolades include being ranked #1 in the nation for social mobility, and our designation as an Innovation and Economic Prosperity University by the Association of Public and Land-Grant Universities. Our research expenditure recently exceeded more than $250 million per year, and our diverse patent portfolio includes something like the seedless tango mandarin, which has transformed the citrus industry in the world. Nobody will eat this day a mandarin with seeds, and that's happened thanks to the cuties that you may find in the supermarket. Today's lecture exemplifies the mission of our campus, and the Jane and Jerome Schultz Lecture Series in Engineering was established to bring outstanding speakers who are world leaders in the bio and engineering fields to Riverside and to the inland Southern California region. The series follow, as some of you may know, a biennial cycle alternating between bioengineering and biology in one cycle and the broader field of engineering in the next, creating a dynamic and interdisciplinary platform that reflects how modern research truly works across discipline at multiple scale and in collaboration. The Schultz Lectures represent more than a distinguished speaker series. It's a powerful investment in intellectual exchange, education, and community engagement. For UCR, it strengthens our role as a hub for cutting-edge research and thoughtful leadership. For our students, it provides direct exposure to scientists and engineers who are shaping the future. And for our region, it brings global expertise and perspective to inland Southern California, reinforcing the idea that world-class research happens here. Since its inception, the lecture has set up an exceptional and high standard. In 2023, the inaugural Schultz Lecture was presented by Professor Eric Betzig. I am the study collaborator of our speaker today, who was recipient of the 2014 Nobel Prize in Chemistry for his pioneer work in super-resolution fluorescent microscopy, research that fundamentally changed how we visualize life at nanoscale. And today, we are honored to continue this legacy with Dr. Lippincott-Schwartz, world leader in cell physiology, whose work has transformed our understanding of cellular organization and dynamics. She will present a talk on looking under the hood of cells from micron to atomic scale. In closing, I'd like to acknowledge and thank everyone attending today, and all of you who have been involved preparing this event, which I know take a lot of work. And with the permission of our speaker, this lecture are streamed online and reach a broad international audience, exceeding the impact of the series far beyond this room and affirming UCR a place in the global research community. Thank you very much.
Okay, thank you, Rodolfo. And the next Dean, Chris Lynch of engineering, he's gonna introduce the role of engineering.
Just a few words, and I will be brief, but thank you, Rodolfo, for the wonderful introduction of the university and the Schultz lecture and how much impact this has. I have been honored to serve as the Dean of Engineering here for the past 8 years through a period of just tremendous growth and real benefit in my own reputation from the excellence of our faculty and the research that they do. One of the highlights of my time as dean has been Jerome Schultz, our former chair of bioengineering, endowing this lecture series that enables us to bring just some real world-class people here to share their thoughts, ideas, research with us. Tremendously important. Just to give you an idea of the growth that we've undergone, when I arrived here, there were 3,400 students. We're now over 5,000 students in engineering. We've grown the faculty from 122 to 154, enabling us to really strengthen existing research areas and expand into new research areas. And with that has come a growth in our research expenditures that have gone from 32 million to 45 million in the college. We're going to see a dip in the next year just with what's going on in the federal government, but with the faculty that we've hired at the both junior and associate level, and the way their careers are taking off, this growth, we're on this trajectory for decades to come. So I'm really appreciative of all that everybody in the college has done to contribute to where we are, to events like this. And Jennifer, thank you for joining us, and look forward to your talk. Let me turn it back to Val.
Thank you, Chris. And of course, this lecture wouldn't happen without Jerry Schultz endowing it. So he is gonna do some presentation of how it led to that lectureship into bioengineering.
I have one, thank you. So, let me get this out. Okay. When we were planning for this lecture, Val suggested that I give a little bit of the history of the department. This is the 20th year of the establishment of bioengineering at UCR. But going back to the beginning of the concept of having a department of bioengineering was due to Dean Satish Tripathi. He was interested in bioengineering, and through a series of interviews and connections, he got in touch with me about joining UCR as being the chairman of bioengineering. The college was somewhat new, maybe 20 or 30 years old at the time. And I was a little bit nervous about the resources available. So then we talked with Dr. Cordova, who was the chancellor at the time, and she assured me that the funds would be there for getting the department ready and growing. So my wife and I joined the university in 2004. I, as a professor actually of chemical engineering as some of the other starting faculty. And Jane was the Director of Research Development in the Office of Research. One of the first issues in starting a department is what is the subject and what's the goal of the department and what's the focus of the department, particularly because there are bioengineering departments all through this UC system. And Jane and I had worked with NASA a couple of years before, developing some ideas of using biotechnology in the Mars mission, and that sort of guided me to come up with an idea for bioengineering here. So what I selected was the topic of biocellular engineering. At the time, some of the other UC departments were in genomics, more in clinical bioengineering, and so I've thought this at the level of the cell would be a unique contribution and a place to grow the department. So some of you may notice that this was a slide that we used to attract faculty and also to attract students and also to publicize the growth of the department. So these are the first faculty to join the department, and they all were initially in the chemical engineering department. Bauman had been doing research on membrane biophysics. Jayu had been doing research on metabolic control and drug discovery. Dimitri had been doing research on protein configuration. Val had been doing research, he invented the term bio-inspired or chemistry, and Victor had been doing research on proteins in concentrated systems. And at that time, the MS&E building hadn't been constructed, but we at least had a draft plan for the building. So that was the basis of our getting it started. Now, oh, I did the wrong thing. I'm sorry. Okay. Oh, sorry about that. But this is the department today. The faculty that were hired when I was here are the little ones with the little stars. But as you see, and as the dean mentioned, and Dr. Ho promoted the department, and now the department has grown actually faster than the College of Engineering has grown. So congratulations to the department, how well it's grown, and we look forward to the next 20 years. Thank you.
Okay, thank you, Jerry. That was really nice travel in the past with some of the pictures that I don't remember they existed. And now it is really my pleasure to introduce the speaker for today, and everybody saw her CV, but her path to where she is now, it can be quite educational for many of the young students. How to develop executive mind and pursuing things at a different level. Although she was always inclined towards science, her undergraduate degree is in philosophy and psychology. And that that preparation gives you really good foundation of the ability to go in intricate detail in critical manner without losing the sight of the big picture. And then, also another interesting fact is, she didn't directly go to graduate school, she went teaching in Africa, teaching science, which is another important part of developing, understanding the world and so forth. And there is a faculty in UCR, he unfortunately passed away, but he was saying that after students finish their terminal degree, maximum degree, they should not be allowed to get a job unless they document that they've been to at least 50 countries around the world. So he almost did that. And then she went for her master's in Stanford, where she studied DNA degradation and DNA science. And that actually started her thinking, why do we need to destroy the cell? It's more interesting to study the whole cell, which is what we'll call systems biology nowadays. That brought her to Johns Hopkins, where she got her PhD. Then she went to NIH, continue on this work and really deepening it. And that's where the collaboration with Eric Betzig started. After you discovered how to use light to control the fluorescence quantum field of proteins, which is, in organic chemistry, we'll call it the photochromic effect. But you actually, on GFP, is a little bit different. And then in 2016, we moved to Janelia and really built an amazing program in cell physiology. Now, it is no wonder she's one of the few people in the nation who's member of two of the national academies, National Academies of Medicine, National Academy of Sciences. Numerous awards, you can read about it. I'm terrible at memorization, I'm not good at that. And today she's gonna basically connect some things that we, I mean, I'm not gonna give away her talk, but if you read the abstract, things that, many of the things that we hear on everyday news, like COVID vaccine and so forth, and how to do really cutting edge science with that. So without further ado, Jennifer, the floor is yours. We need to switch the computer. Oh, yes, yes. Oh, before we go into that, the most important part, sorry.
So this is just recognition and a thank you, and yeah.
Thank you so much.
So, and there's a cover to the box as well. And you have Jerry,
Photo op.
come over, just, and Val, you too. Come over for a photo. That's great. Val, let's get you in the picture. Yeah. There we go. All right, thank you. And I'll hold this for you.
We should try the other one. Yeah. Yeah, okay, perfect. Let's put this in here instead. So you can hear me? Yes, I want to thank the organizers of this, including Val, for allowing me to come here today. So I'm going to be talking about some technology. My title is slightly different than what was on the program. The title is Harnessing mRNA to Illuminate the Hidden World of Cells. Now, we're going to look at micron scale processes, but I'm going to be sharing with you a new technology that I think, what's sort of it's an old technology, but we're using it in a way that I think can be extremely important for being able to visualize processes within tissues and whole organisms. So to put everybody on the same page, because I don't know how many people here are actually doing imaging as opposed to biochemistry or molecular biology, but imaging life, imaging biology has in fact been a very long-standing curiosity among humans. Basically back in 1670, Van Leeuwenhoek really built the first microscope and revealed that there's a whole world of activity going on that we can't see, but we can if we have improved technology in the form of lens and microscopy. And this just brings you up to date here in 2026. We now have these incredible instruments developed by Eric Betzek as well as other engineers called lattice light sheet microscope, which allows you to be able to look at individual cells and huge populations of cells within tissues at really exquisite time and spatial resolutions. Now, what is unique about studying cells by imaging is that it allows you to really do three things. It allows you to study events up close, so you've got space. It allows you to follow rapid changes. These microscopes are real time. Basically, you're studying a process, which we weren't able to do in the old times. And then finally, it allows you to manipulate an experiment, to control your sample. You can look at, what happens, as a sample gets desiccated or a sample gets stressed, changes in nutrient, et cetera. You can manipulate this system very easily and look at the response by the cells by imaging. But despite this, there are many challenges to imaging cells that over the last 200 years has required innovations, not only in the microscopes, but labeling strategies so that you can actually see things within cells, and also conceptual frameworks. When people first looked at cells, they had to articulate what a cell is. When Van Leeuwenhoek first saw a tissue under his microscope, we didn't have the term cells. So the concept of cells had to be envisioned. And so in the course of sort of the history of imaging, there's a lot of integration of all of this happening. When you see something that nobody else has seen before, you need to label it, you need to put it into a conceptual framework. Now, in my lifetime, a huge impact that had a huge effect in the system was labeling strategies, and in particular, labeling strategies made available by the green fluorescent protein. GFP is a protein found in this jellyfish that will glow in response to light. And the key excitement with GFP is that it works all alone. I mean, basically, this molecule itself, you shine a light on it, it will give light off in a different wavelength. And because of that, you can engineer any protein that's within a cell, tag it with this GFP, and you will make that protein fluorescent. It will light up. And so as a consequence of that, it transformed the field of cell biology because you could basically now visualize any protein, any structure within the cell using this GFP as a tag for that structure. Now, from 1995 to 2026, these are all the imaging technologies that I myself have used or helped develop in some way. There's many other imaging technologies. Many people have been involved in this sort of flow of sort of evolution of these imaging technologies that were really fundamentally made available by this green fluorescent protein. And Val mentioned PALM, which Eric Betzik's single molecule imaging of these fluorescent proteins that was developed. Well, despite all of this, despite its successes, the delivery of GFP is only possible in cells that are dividing. Many cell types, the ability to get GFP into those cells is extremely difficult. And those include neurons, immune cells, muscle, hepatocytes. The only way you're going to be able to visualize GFP in these cells is through introduction of a virus that will deliver the gene of interest. Even then, it's extremely tricky and takes a very long time because you have to get that virus integrated into the cell. And so it's made it extremely difficult to use all of the technologies that I just mentioned to look at what's going on in cells that are really important. The vast majority of the cells in your body are actually non-mitotic cells, very difficult to express a GFP in. So we were wondering whether we could get around this in some way by harnessing the power of mRNA technology. And we're not the first to think about this. In fact, it's this mRNA technology really that has been pioneered by many, many people. But I want to say that all of the COVID vaccines would not have worked without mRNA therapeutics, in particular therapeutics that alter the mRNA to make it more stable within a cell when it's expressed in the cell and less prone for the mRNA to trigger an immune response. And in fact, the Nobel Prize in Medicine in 2023 was given to Carrico and Weissman for their discovery regarding nucleoside-based modifications in mRNA that enable the development of effective mRNA vaccines against COVID. If you did not modify the mRNA in those vaccines with modified nucleosides, those mRNAs would trigger a massive immune response in your body, and that vaccine would not be very helpful to you. So it's only with this discovery and this implementation that we had COVID vaccines. So it was a breakthrough not only in vaccine implementation, but also it made the field extremely interested in how you could modify mRNAs to make them more stable within cells so that they could express proteins of interest. So Hee-Jin Choi, incredibly talented postdoc in my lab, decided to see whether we could use these modified mRNAs as a universal gene delivery platform to allow us to begin to visualize any type of protein or biosensor in these very hard to transfect cells. So we're going to be leveraging the advances from mRNA therapeutics to see if we can figure out how to get proteins of interest into these very hard to transfect cells. And so basically, we start with optimizing protocols for in vitro transcription, where we're going to make stable, low toxicity mRNAs with a gene of interest that we're going to be visualizing. That gene of interest is going to have a GFP associated with it, so we can visualize it once it gets into the cell. Now, we're going to try to screen different modified nucleotides to make sure that when this mRNA gets into the cell, there will not be a massive sort of immune response. This mRNA will be stable and will not trigger stress response pathways. And so these are the different modifications that we're going to be looking at in these mRNAs. One is a cap sequence, which is important for the RNA to be stable in the cell. And this is the pseudouridine, which is what was originally used in the Nobel Prize groups in our vaccines. This is M5C that previously people have found also can help stabilize mRNA within cells. And this is 5-methyluridine. So we're going to look at each one of these modifications and compare how they work when we transfect, when we put an in vitro transcribed mRNA with one or more of these mutations, mix it in a tube with lipofectamine, which basically is a delivery system that we can then transfect the cells with. Pretty straightforward. So these are the 9 different types of in vitro transcribed mRNAs that we're going to be testing to see, is there a universal modification code that will allow us to be able to introduce any type of protein of interest. And so these are the different, this is the symbols, this is the modifications that are going to be put on that mRNA. So we start with U2OS cells, which are commonly easily transfectable cells. Our goal here is just to see which one of these mRNAs that are modified with these different nucleotides modifications is going to give us better expression. So here are the cells labeled with DNA, and this is a nuclear M scarlet. Scarlet is a type of fluorescent protein, and it's targeted to the nucleus. We have a nuclear target to it, and now you can see how efficient the mRNA that's being delivered through this transfection is. And I think you can see pretty quickly, it's these 3 mRNA species modified either with a 5' cap, a methyl uridine with the cap, or the M5C with the cap that give the best type of transfection in this cell population here. Now, we know from the literature that the 5', well, first off, it's only mRNA that have 5' caps that will, once the mRNA is delivered into the cell, that the mRNA will be stable. So basically, it's only, all of these guys have the 5' cap. What's different about them is whether it's the uridine or cytidine modification. What has been found with the COVID vaccines is that this modification right here, which is being used in the COVID vaccines, actually can give rise to frame shift mutations. And so they may not be the best way to introduce your gene of interest. So we decided to, and the same is true with this 5' cap alone. So we decided to focus in on this 5' cap M5C modification and look to see what its impact is in hard to transfect cells. So here what we're doing is we're taking this modification here, which is right here, and we're looking to see how well it, when we transfect it in with a nuclear targeted protein that's fluorescently tagged, how well it expresses in all of these different cell types. Normally, a cDNA would hardly, you'd only get a small fraction, if any, in each one of these cells if you did a DNA transfection in hepatocyte, muscle, neuron, macrophages, because they don't divide rapidly, and certainly limited in the organoid. But we're seeing all of these different cell types and populations are transfected very well with this M5C RNA species. And this is just an example here. These are hepatocytes grown on cover slips, and you can see with the nuclear targeted fluorescent protein that has the mRNA encoding this nuclear targeted sequence that has this modification, it goes in extremely well into these cells. If there's no 5' cap on that mRNA, you don't see the transfected species. We can also look at whole brain organoids. So this is a brain organoid. This is the DNA in that brain organoid. And if we pipette onto that brain organoid these mRNAs that have the 5' cap M5C, you see all of the cells light up, indicating that we have the ability to visualize proteins in this system. Now, what's exciting is that if you look at the time course for expression of the mRNA in these cells, we see it within 3 to 5 hours. Normally, if you do a cDNA transfection, You're lucky if you see something 24 hours later. We're seeing within 3 to 4 hours transfection. And it's the majority of the cells. So here is an ER, a mRNA expressing a protein that's targeted to the ER, Sec61 beta. So you're lighting up all the ER in these cells. And here's a nuclear targeted mRNA. This lasts for long periods of time. So it's a fantastic system for immediate expression of these proteins. We can also not just look at proteins that are targeted to the nucleus or membranes. Here are structural proteins whose mRNA we introduced into a rat astrocyte or rat neurons or a human macrophage, including, this is the microtubule associated ensconcin, targeted ensconcin. So these are microtubules that we can light up in these astrocytes. Here is actin that we can look at in the rat neurons, and here is vimentin, which is an intermediate filament that we're seeing in human macrophages. Previously, it would be extremely difficult to see these structural components in these systems with cDNA approaches. We can also introduce biosensors. This is GCamp, which is a calcium-sensitive probe. It lights up in the presence of calcium, and you can see in a brain slice that we've transfected with the mRNA encoding this calcium sensor. All of the neurons take up that sensor, and you can start seeing calcium signaling in the brain slice. And here's a cardiomyocyte that is induced pluripotent from an induced pluripotent cell, and you can see this cardiomyocyte that's expressing that GCamp sort of flash when it is responding to calcium regulation. And here's just a quantification of the various frequencies of those calcium flashes that you can monitor in these systems. Now, one of the things that Hughes really quickly started realizing is it would be really, can we express more than one mRNA that's coding a particular protein at the same time? And the answer is yes. If we take 4 or more of these mRNAs that are encoding different proteins tagged to different fluorescent proteins that glow in different wavelengths, we can combine all those mRNAs into our lipofectamine mixture and basically transfect cells via this lipid nanoparticle approach. And we call this mRNA bow because we're going to get a rainbow effect. So here's a cell, here are cells that were transfected with 4 different mRNAs encoding different fluorescent proteins that allowed us to see each of the different fluorescent proteins. Now, in this case, each of these fluorescent proteins were tagged to a nuclear targeted sequence. But we can tag these mRNAs to different places in the cell. And that's what you're looking at here. These are adult mouse hepatocytes, and we have an mRNA that's targeted to lysosomes, targeted to the ER, targeted to the mitochondria, and targeted to peroxisomes. And we can see the distribution of these organelles in the same cell as a consequence. And this is in human primary macrophages, the same thing. Here are lysosomes, ER, mitochondria, and peroxisomes that all light up with different colors through this mRNA bow approach, which is really simple. You know, you're just putting all these mRNAs in a test tube that have and then you put lipofectamine to put them in a lipid nanoparticle, and then you just pipette in that mixture on top of these cells. They take up the nanoparticles just as, you know, in your Moderna vaccines, they're taken up by your cells, and you express these proteins. So we can also see this mRNA bow work at the tissue level. Here's a brain slice, and we're introducing 3 different mRNAs expressing different organelle targeting sequences, and they have different colors so we can identify them. So we can see how they light up the whole brain slice. And this, if we zoom into single cells, you can see the distribution of ER, mitochondria, and lysosomes in these single cells within the brain slice. So then Hughes said, okay, well, let's see if we could get it into a whole organism. And so this is what we're going to do is we're going to inject this in vitro transcribed mRNA that's carrying the fluorescent protein and the protein, targeted sequence at the single cell stage in an embryo. And I'm going to start with zebrafish. So here is a zebrafish where we have injected a nuclear targeted fluorescent protein at the single cell stage. This is 4 days later, and you can see every single cell in this zebrafish is expressing this fluorescent protein in the nucleus. Now, the reason why this is so colorful is that we color-coded based on depth. We're just Z-sectioning through the depth of this zebrafish, and we color code based on the depth so that you can see the whole depth through this zebrafish. Importantly, we can start introducing biosensors. In this zebrafish, we're at the single cell stage, we're going to introduce this biosensor, GCAMP6, to try to look at calcium fluctuations in all of the cells of this zebrafish. And we're focusing in right here, which is in the brain. But you can see these flashes that are periodic along these cells represent hotspots for calcium fluctuation. In this system, we can also introduce the mRNA bow. So here we have 4 different organelle markers that we've introduced by different mRNAs that are going to be injected at the single cell stage. And you see 3 days later, all of the cells in the zebrafish are expressing these mRNAs, the proteins for these mRNAs that allow you to see peroxisomes, mitochondria, lysosomes, and ER. And if we can also do this in other organisms. This is Janelia. It's another type of fish, which is particularly interesting because this fish doesn't develop pigmented skin, so it's easy to see this the development of this fish through adulthood. And again, in the Janelia, this is 3 days after fertilization, you can see the development of that fish and how all of these different organelles are distributing in different cell types. And this is just zooming up here. I don't know if you guys think hard about these organelles, but they are playing very important roles in the metabolism at the single cell level in this system. We can also introduce these mRNAs into non-model organisms that really are very hard to transfect with cDNA approaches. And one example is the sea urchin. This is in collaboration with Jia Song at the University of Delaware, where she's very interested in the sea urchin life cycle. And when we talked to her at the ASCB meeting and we told her of our approach, she was super excited to inject these mRNAs at the single cell level and watch sea urchin development vis-a-vis the visualization of the cell populations and the way organelles are distributing across the cell. So this is at the gastrulation site. This is 48 hours after injecting these mRNAs. And you can see all of the cells surrounding the gut are lit up. You can see precisely the distribution of the mitochondria and lysosomes in all of these cells. So we're super excited by the potential of this mRNA bow to basically allow us to take fluorescent proteins as well as halo tags and introduce them into hard-to-transfect systems, which include neurons, immune cells, hepatocytes, muscle, organs, and whole organisms. And that will allow us to begin to really dissect cell biology of these systems, of these organ as well as organismal systems in ways that we have not been able to do at this point because of the difficulty of the cDNA expression approaches. Okay, so, and I just want to say this work has come out recently on bioRxiv. We're very happy to share with you these mRNAs, so feel free to contact me if you want to learn more. And bioRxiv it describes how we make the mRNAs, et cetera. But for the rest of my talk, I want to continue with the discussion of mRNAs and their ability to be used as tools to probe cell biology. And I should just say, when I started in this field of cell biology, I was just fixated on proteins. You know, I really ignored the mRNA field. But mRNAs are really important tools that really can help us unravel a lot of different processes. Okay, so what is it we're going to look at? So we want to visualize these mRNAs within cells. What are they doing? How do they move around in the cytoplasm, et cetera? And fortunately, there's some technology that's already available, developed by Rob Singer and colleagues, where you can modify the mRNA with some stem loop structures that will can serve as a binding site to a fluorescent protein that's tagged to a coprotein that binds to these stem loops. And when you do that, you light up the mRNA. And so we're going to look at this. We're going to use this system to probe protein synthesis. Basically, here's an mRNA that when it's fed through a ribosome will produce a protein. We're going to watch this process. We want to watch this process using these technologies. Now, we're going to focus on a particular class of proteins that are being synthesized. And these are proteins that encode membrane and secretory proteins, which represent 30% of the genome. The reason why we want to look at these guys is because they are known to be synthesized on the surface of the endoplasmic reticulum, on the surface of a membrane, because ultimately they're attached to a membrane and they've got to be pushed through this membrane to enter the secretory pathway. And so all of the ribosomes that are doing that translation, we know are associated with the surface of the ER. This is an EM image. Each of these dots represent ribosomes, some of which may or may not be undergoing translation. But what makes this especially interesting is that the proteins that are translated on the ER they have to be targeted to the ER, and then that translation is coupled to a translocation of the protein, the newly synthesized protein, through the membrane of the ER. And then it gets these proteins either embed here and then move through the secretory pathway to localize to different organelles, or they get into the space of this ER where they get secreted through the secretory pathway. But all of these proteins come from mRNA that gets pulled to the surface of the ER through this pathway, this co-translational translocation pathway. Okay, now why do we want to look at this? Because we're very interested in where this is happening in cells. It's happening in the ER, but you can see this ER that we've highlighted here using fluorescent imaging technology is extremely complicated. Where on the surface of this complex meshwork is translation of these secretory membrane proteins happening. Here is only on specialized parts of this ER. By visualizing these mRNAs that are in the process of translocating, we might be able to answer that question. So in order to do that, Hughes Choi in my lab developed, basically generated an mRNA that is able to be labeled with a halo tag, but specifically mRNA that is that's encoding membrane and secretory proteins that are going to dock onto the ER membrane to translocate. He then expressed it within cells and visualized these mRNAs, we're calling them secretome mRNAs. So this is the cell. All of this sort of web-like structure is the ER, and each dot is a single mRNA that is encoding a secretory membrane protein. In this case, it's cell transferase. So you can see how these mRNAs are moving around in the cell, and they look like they're binding all over the place. But Hughes went in, and he realized very quickly that there are two pools of these mRNAs. Some are fast-moving, which we've color-coded pink, meaning once they're on the ER, they move quick, and then they fall off the ER. And others are slow-moving. They're attached to the ER, and they don't move much. They're confined in their motion. And so that made us think, well, is it possible that the slow-moving mRNAs are translated? You would think that if you're translating on the surface of the ER, that process of shoving a nascent peptide through a membrane may retard your mobility on the surface of that ER. So how do we test this? The first thing we did was we treated with this drug called puromycin, which blocks protein synthesis. And by basically preventing translation. And when we did that, we converted all of these mRNAs into very fast-moving mRNAs. They're unable to translate, and they're no longer showing this type of confined motion on the surface of the ER. So that said, okay, cool. So these slow-moving mRNAs are likely to be translating, but can we prove that? And in order to do that, we made use of another technology that allows you to visualize an mRNA that is in the process of translocating, in the process of making a protein. How do we do that? Engineer an mRNA that encodes a GCN4 epitope. It's got many epitopes here, and what this epitope can do is bind fluorescent nanobodies. So we can light up, as soon as this epitope is translated, the nanobody that's fluorescently tagged will bind it, and we'll get a signal. And so we'll see a signal from the mRNA, and we'll see a signal from the newly synthesized protein in the same place. So we know then that this mRNA is translated, okay? So when we did that, what we found was when we have mRNAs that don't have this sun-tag signal, those mRNAs are jiggling all over the place. When we see an mRNA that has a sun-tag signal, this is single particle tracking. It's not moving. It's highly confined. So this is the behavior of a translating mRNA on the surface of the ER, and this is one that is not. So that proves that when these mRNAs are translating, they have highly confined motion. So that then led us to say, okay, well, let's look at where the mRNAs are localized on the ER. Is there anything special about where they're showing the confined motion on the ER? And what we found was mainly these guys are at junctions. They're at sites where this ER is coming, basically touching, forming a cross-linked meshwork. They're at the hubs of the meshwork. And we see this not only for, we're looking at 4 different mRNAs that are encoding 4 different types of membrane or secretory proteins, and all of them are showing this highly confined motion at the ER junctions. So, and this is just showing, we can quantify this looking at the distance from an ER junction. All of the membrane proteins that are being translated are very close to those junctions. Actin, which is not translated on the surface of the ER, is nowhere near these ER junctions. So can we look at this using other approaches? So this is volumetric focused ion beam scanning EM. So we're now at a whole nother level of resolution where you can see much more detail that ER. So in blue is the structure of the ER that we've segmented out from these EM images. And in green here are ribosomes that you can see on the surface of the ER. So we've segmented and annotated the ribosomes that are associated with the ER. And when you do that, you can quickly see that the ER ribosomes there's a concentrated pool of ribosomes at these junctions. We just zoom in here. Here's one ER junction. You can see a whole bunch of ribosomes here. And what is really interesting is that many of these ribosomes are lined up in a pattern that is very reminiscent of what we call polysomes. Now, I know a lot of you are not hardcore biologists, but a polysome is a single mRNA that has many ribosomes that are simultaneously translating protein off that mRNA. And people have shown sort of ribosomal arrays like that. And we're seeing a ribosome array here, a ribosome array here. So our thinking is this is a single mRNA with a whole bunch of ribosomes associated with it. And that would be indicative that this pool of ribosomes is, in fact, translating, supporting what we saw with the SUNTAG system. Okay, given that, the question is, okay, you know, as a biologist, you go mechanism. What is making these ER junctions hotspots? Well, intuitively, you could say, well, they're a flatter surface. It might be easier to form a polysome there. But is there any other important pathway or process that's critical for this? And we are organelle biologists in my lab, and so we started thinking maybe there are other organelles that are associating with the ER that could help in the translation. And so we focused on lysosomes. This is focused ion beam imaging of a lysosome where you can see it's closely associated with the ER. We can also take advantage of a microscope, this grazing incident turf structural illumination microscope. Unbelievable. It was developed in our physics lab. Unbelievable in terms of its speed as well as the super resolution that you get. But we can visualize ER in pink, and these lysosomes are green. And you can see how intimately associated the lysosome and the ER are. They love each other. I mean, the lysosome is just sliding along the surface of the ER, but frequently it gets trapped at these junctions, sites where ER are coming together to form these junctional regions. So that led us to ask, is it possible that this lysosome, which we know is playing a really important role in cells in terms of providing, basically amino acids and other things, could that be important for translation? And so he used his SUNTAG mRNA labeling approach to see whether translation, which we know we can read out with SUNTAG, of these secretory and membrane proteins is happening near lysosomes. And the answer is yes. Here's a merged image of a translating mRNA that's got the SUNTAG signal vis-a-vis the lysosomes in that cell. And they are right next to each other. That was pretty exciting when I saw that. In fact, if you plot the distance away from lysosomes, you see the SUNTAG signal is incredibly high when the mRNA is closely opposed to that ribosome. And this mRNA it's translating on the surface of the ER. We further found that if we inhibit lysosome function, we diminish the translation fraction of these secretome mRNAs. And we can do that either by neutralizing the lysosome with chloroquine or inhibiting its proteases. So what does this suggest? Well, what it suggested to us initially was, here we got this ER being made. When an mRNA translates a protein, what is it doing? It's grabbing mRNAs and it's grabbing amino acids and linking them together. So it needs amino acids. And in the case of the lysosome, it's a ready pool of amino acids. Basically, to test that, we look to see, yeah. Oh, okay, good. Sorry, I won't walk away. Yeah.
No, you can walk away.
Okay. Yeah, that's a good idea. One amino acid, so we look to see, let's make the cell super dependent on lysosomal amino acid release from lysosomes. Well, how would you do that? Well, let's not feed cells, let's get rid of amino acids from the medium. When you get rid of amino acids from the medium, you're completely dependent on lysosomes for getting amino acids. The lysosome takes, basically, triggers an autophagic, the cell starts doing autophagy, which draws proteins into the lysosomes. They get broken down into the amino acids that then can be released from the lysosome. And when we do amino acid starvation, we see massive sun-tag signal close to the lysosomes. So we are basically making translation very dependent on those lysosomes under amino acid starvation. So that led us to the question of how are lysosomes and ER junctions drawn together. And we did some analysis there. This is a protein called lunapark, which we're going to show has an important role. Lunapark integrates into the ER membrane. It extends off that ER membrane into the cytoplasm, and it turns out that it localizes, this protein normally localizes at three-way junctions. In fact, that's the reason why we started focusing on that protein. It's an ER protein at three-way junctions. Could it be involved in linking lysosomes to these junctions for the purpose of facilitating translation? And supporting that, if we do proximity ligation assays, we can see that lunapark will basically co-ligate with LAMP1 on the surface of the lysosome. We can also see with antibodies to lunapark that it will, the three-way junctions where lunapark is localized are junctions where we can see translation happening using our mRNA and sun-tag system. We can further show that if we knock down lunapark, we no longer have a distance dependency of translation vis-a-vis the lysosome. So our thinking is that lunapark is helping to draw lysosomes next to the ER at these three-way junctions that then can facilitate amino acid release from the lysosome in a localized area that can facilitate translation. And consistent with that, lunapark knockdown abrogates that system. And furthermore, it reduces overall translation of these secretome mRNAs. So here is a particular type of secretome mRNA, encodes acetyltransferase. When we knock down lunapark, you can see the translation fraction drops dramatically. We can also see a global effect in cells. If we just do biochemical membrane fractionation of newly synthesized proteins, you get a drop in that pool when we knock down lunapark, in contrast to cytoplasmic whole cell fraction of proteins, which are not impacted by lunapark. So that led us to ask the question, is there something very special about the lunapark knockdown that's impacting the efficiency of translation of these secretome mRNAs that are translating on the surface of the ER? And the first hint came with seeing an increase in phosphorylated EIF2 alpha, which is an initiation factor that's critical for the initiation of translation. And sure enough, with a Luna Park knockdown, basically you get the reduction of the sun tag signal. If we remove the 5' UTR, that is basically the part of the mRNA at the 5' sequence that is involved in initiation, that is regulated by this stress pathway. If we remove that, we no longer get this regulation by Luna Park knockdown. Furthermore, if we add a drug called ISREB, integrated stress response inhibitor, to inhibit the integrated stress response pathway, we can abrogate the effect of Luna Park knockdown, suggesting that when we knock down Luna Park, we're triggering an integrated stress response pathway. And that's why we see the reduction in the translation. And this is my final slide, just showing through visualization techniques the impact of this Luna Park knockdown on the efficiency of translation. So here is an assay where we're focusing in on one of these sun-tag mRNA co-localized signals. This is an mRNA that is in the process of translating because it's got a sun-tag signal. We're now going to photobleach the sun-tag signal, the nascent peptides that have fluorescence associated with them, in order to see how quick it returns as a result of the continued translation on this polysome. And you can see within 100 seconds it recovers. But when we do this in a Luna Park knockdown cell, we get very perturbed recovery of this sun-tag signal, indicating that after the bleach, this initiation, basically there's the initiation of translation and potentially elongation process itself is all perturbed with Luna Park knockdown. Now we think this is due to a triggered integrated stress response pathway, and consistent with that, if we override that stress response pathway with this inhibitor, we override this inhibitory effect. So I'm just going to sum up what all of this means as far as we're concerned. We think that using these mRNA tools to be able to visualize protein synthesis, we found that for membrane and secretory protein, that protein synthesis is happening at these junctions predominantly. And it's due to, we think it's facilitated by the fact that lysosomes can be drawn to this area through Luna Park. The lysosomes could enhance the translation through directed amino acid release. Also, we think on the surface of these lysosomes, there's tRNA synthetases that can help charge these amino acids so they can participate in translation to facilitate that pathway. But I want to, I'm happy to answer questions related to this project, but I know we're running late, so I want to end by just thanking people in my lab, as well as Janelia, for really the opportunity to do this kind of work. We've been tremendously benefited by just the whole Janelia environment. I want to shout out Hughes and Choi, who did the majority of the work that I talked about today. But again, we were benefited tremendously by people like Eric Betzig, Harold Hess, people outside contributors who have also helped in this whole process. Thank you so much.
Okay, thank you. So now it's open for questions.
Incredible talk. I'm going to take some notes on how to give a presentation from you. I was going to ask, though, so why does it need the junction? Why can't it just do it away from the junction in terms of protein synthesis?
So we're not saying that you can't do any translation at all from those junctions, but we're saying that the availability of those junctions has enabled the translation to be more efficient and more controlled. We think that the junctions, by virtue of the fact that the lysosomes are so intimately sort of moving on the surface of the ER, and they get associated at those junctions, potentially via Luna Park, The lysosomes are providing an amino acid pool to make it easier to build the polypeptide during translation. If you don't have the lysosome right near where the translation is occurring, our data would suggest that it's not as efficient. And you can trigger an integrated stress response as a result of that. In order for a protein to be able to push itself through a membrane bilayer, you really need to have a very regulated, the system needs to be much more regulated, we think, than translation of a cytosolic protein, which is floating around in the cytoplasm. So we think that this is it's a highly dynamic system that is making use of the fact that you have these lysosomes that are ready pools of release of amino acids that the cell can take advantage of for really tightly coupling translation and translocation on the surface of the ER.
Thank you.
Yeah.
Thank you, Jennifer. That was great. So is translation blocked or diminished in lysosomal disorders like in Gaucher? Or would you expect that to happen?
Yeah, we haven't looked at that. I mean, it's a really good question, but yeah. I mean, there are, I frequently get asked the question of, is there a specific type of lysosome that is at these three rate junctions? Presumably it's a lysosome that is acidic, that's breaking down amino acids, so it would be very interesting to see, whether all lysosomes or just a subpopulation are participating in this.
Thanks for the fantastic talk. So I'm thinking about the application of the mRNA technology for GCAMP or channel rhodopsin delivery, like in live animal. So have you integrated these mRNA with, let's say, CRE-LOXP kind of like to deliver for transgenic, animal for a specific
I mean,
cell
could we
type?
use that technology is what you're asking?
Yeah, is it available? Let's say, you have these DIO, tags that you make it so that by injection in the Cree animals we can, because
Yeah,
this is going to
yeah.
be very helpful. We don't need to wait 4 weeks, 5 weeks, after the
Yeah,
it's
no,
not,
it-
makes everything.
Yeah. I think we're just at the very early stages of how to use this technology in different ways to turn on or off genes at different stages in development. Yeah, we're interested in trying all kinds of
the
stuff.
Cree lines. And the second question, have you thought about like specific for the neuronal application, for the retrograde kind of application, so that these mRNAs are kind of picked up by the axonal terminal, and then they go back to the cell bodies. So similar, we have the retrograde virus for the AAVs.
Yeah, yeah.
So is it possible for this, to add more specificity for this?
I mean, yeah, I mean, why not? Basically with the AAVs, it's just a hit or miss. It's random which of the neurons are taking up the AAV, and so you see, low-level integration across the system that you've transfected.
But like for this retrograde virus, they are only picked up, let's say, by axons. Not
Yeah,
by
yeah.
axons. So is it possible to make it only picked up
By an axon? Yeah, so we
Terminal,
haven't
at the
tested that
terminal.
at this point, but
Thank you.
definitely could try.
Other question? Yeah, go ahead.
It was a really good lecture. I thought it was really great how you explained everything sort of chronologically. My question is so what are the next steps? Would it be why are these Luna Park proteins attracted to these ER junctions? So it's like, I see it as like it's sort of a chain where the ribosomes are attracted to the junctions because the lysosome is there. The lysosome is attracted because the Luna Park proteins are there. What's attracting the Luna Park
Yeah,
proteins?
what's the chicken or egg? Who goes there first?
Exactly.
Well, when we image these mRNAs, we can see that initially they're diffusing on the surface of the ER, and then we can see them get sort of confined. And the thinking is that they start getting confined because maybe this junction is a larger platform for the ribosomes to start engaging a single mRNA like this. But subsequent, I mean, the association with lysosomes has really puts a boost in this system by increasing the availability of amino acids. But you know, you could argue, well, maybe it's the lysosomes that are here first, and the mRNA comes and it locks in here because it can start translating. So it could be either. I don't know. Yeah.
Maybe the translation of the Lippincott protein can come from the same process where these polysomes are also not only creating secretory protein, but also the Lippincott themselves.
Yes.
It's like this
It's a cycle. Yeah.
cycle.
Yeah. That's probably.
But
The way
something
it
else
is.
from geometry point of view, is it possible that Luna Park is everywhere, just the junction allows that to bind from multiple sides, so then you increase the binding with the lysosome, because lysosome is...
Yes. Yeah. Yeah.
So basically, it's a geometry and multivalency
Yeah.
way.
Yes.
The lifetime of bound
Yeah.
lysosome is longer.
And there could be other proteins on the surface of the ER that are helping to stabilize this interaction.
Other questions, Jayu?
I think it's a great talk. You know, so the imaging now, and they just look at where the molecule is, but they also solve some kind of mysteries in cell biology. So what is special of that nucleotide, the 5-methyl cytosine? Because that makes an mRNA very stable in the different cells. Because in some cells are very difficult to transfect. I know. That's the first question. Second question is, during this cartoon, where the tRNA synthetase is located? Within the lysosome
Yeah.
or
So
just outside?
I'll answer the first question, the last question first. It's super interesting that, and we've supported it, that some of the mRNA, the tRNA synthetases are on the surface of the lysosome. They are, which makes perfect sense, that they are right there ready to meet the amino acids that come through the transporters, charge those amino acids so that they are available for protein synthesis. But not all of the tRNA synthetases are on the surface.
So that's interesting, because that's a special class of tRNA synthetase, or because the lysosome digests everything, right, supposedly?
Yeah. So that's something that we're looking at right now. Membrane and secretory proteins are enriched in leucine as well as asparagine due to, in order to draw them across the bilayer. And one of the tRNA synthetases, I believe, that is associated with the lysosomal surface is tRNA for leucine. So that would fit with that idea. But that would be fantastic to really look into.
Okay, Sean?
Thanks for the fantastic talk. I was just curious, for the maintenance of those polysomes at the junction, is that, from what you see, is the balance of that reinitiation of terminating ribosomes, or is there a good deal of de novo initiation from ribosomes being recruited away from the ER?
Yeah, maybe, can you just repeat that question?
Yeah,
again?
sorry,
You
yeah,
wanna know
so the polysome is there, and you have this wonderful imaging and the ability to track it in real time.
Yeah.
And so, there's always this question of whether initiation of translation, what extent of it is reinitiation of ATS particles that get recycled and pushed back to the 5' end versus...
the translation rate itself.
Yeah.
We can't, so I think you're referring to this experiment right here. We cannot distinguish the difference in kinetics that we're seeing in recovery between control and Luna Park, whether that's due to a change in initiation or elongation rate of the polysome that we're looking at here that's undergoing this translation cycle. We think that it's both at some level that's being impacted, and our data related to the effect of ISREB, the integrated stress response coordination is at the 5' end. It's going to be initiation. So we think initiation is playing a huge role here.
Thank you very much.
Yeah.
Other questions? Right there.
Thank you very much for the great talk. I just have a question related to the polysomes. I'm wondering that the number of ribosomes a polysome carry has any correlation with the size of the ER junction around
Yes. So there's,
it? Thank you.
we're seeing the closer the polysome is to the lysosome, the more ribosomes are associated with it, because we're seeing a brighter SUNTAG signal. So the closer that mRNA gets to the ribosome in this, so the idea would be that once you get that lysosome close here, we're seeing more of these ribosomes on this mRNA. And you're getting, because we get much more SUNTAG signal. So they're translating more actively.
So the structure of the polysome would vary as the lysosome get closer and closer to the ER junction.
Yeah.
Okay.
Yeah.
Thank you.
Right. Other questions? Oh,
It's
gosh.
great.
That's right. We should have more than one microphone.
No, don't throw it.
Excellent talk. Oh, excellent talk. I had a question kind of linking in my mind the first part of your talk and the second part of the talk. I really love this incredibly elegant, the lysosome associates not just with the ER, but with the junctions of the ER. Going back to the first part of your talk, you have this very elegant tool for imaging 4 and I believe even more organelles at once. Can you do some sort of hypothesis generation work where you just look at the structure and see what associates with what at different points in time, and use that to kind of drive, well, there might be something going on here? Can you use that structural information to kind of guess what might happen?
Yeah, I mean, well, first, this is a different scale than the first part of the talk. I think what we're excited about in the first part of the talk, where we can have multispectral imaging in all of these different cell types that we hadn't seen before, we can start asking, what is the position of lysosomes relative to ER? What is the position of mitochondria? We haven't been able to do that. And so it just gives us a bigger sort of perspective for how this is operating at in tissues and whole organisms. All of our imaging was done in tissue culture cells, but now we can do this kind of thing in a whole organism, in a tissue, and in cell types that we haven't really looked at, to see if these principles are conserved.
I'm really excited to see where that goes.
Yeah, yeah.
And before, okay. So would you read the questions? Yes? So we have a few questions from online.
Oh, okay.
This is from our chair, Dr. Hu. Jennifer, thank you very much for the great talk. We have identified some candidate magnetic reporter genes. Would the mRNA help us deliver the magnetic reporter genes? My student, Vala, will be meeting with you this afternoon. Maybe he could ask you more about that.
I mean, actually, on my conversations with people already, we've been talking about that. Yeah. No, absolutely, it would be unbelievable to put those basically the MRI sensors, magnetic sensors in here. Yeah, that would be through this mRNA technology. It's very possible.
We have a second question. What is the half-life of the transfected mRNA?
It depends on the different cells, the cell type. In a tissue culture cell, like a macrophage, it typically lasts for about 14 hours or more. But in the case of the zebrafish, we had those mRNAs lasted for, we saw the protein expression for more than 2 weeks. in the case of, we had many days in terms of the sea urchin cycle. So it varies depending on the system that you're looking at.
Okay, and I have a third question. Injected mRNA will stay in tissue because they are stable, which means they might not be regulated by the central dogma and would compete with the native mRNAs for the intrinsic expression. Would this cause overexpression leading to disease?
Yeah, no, that's a really good question. We don't know. I mean, it is something, the issue is how much are we overexpressing relative to the endogenous pools of the proteins that we've sort of introduced with the GFP tag. Right now, we don't think it's a lot, but we need to test that. And this is going to be really important if you're trying to introduce something that's going to help, do any kind of replacement therapy, or if you want to, poison the cell in some way. We're going to have to understand exactly how much protein is being delivered through this approach relative to the endogenous pools.
All right. Thank you. Any other question? Oh, Victor?
My apologies, I'm a chemical engineer, so I don't know very much about biology. But I want to go back to your mRNA stuff with the 5'.
Yeah.
And you had 5' coupled with other pieces. And I saw how in your slide you had it where it looked like 5' was the legitimate one, it calls it. But then you added the other two, and someone got it. But when you added all three, they just failed completely.
I know.
Except for the mitochondria. Why is that? When you go to these, go back to
no, there was no, there was nothing special about mitochondria.
Can you go back to the slide after? Yeah, go back to the one after it. It showed like it was bold with the mitochondria by itself, but the triple case.
This thing?
No, you had a list of organelles you were looking at.
Yeah.
And you had the. You go back. It was actually it was a table.
Go earlier. Yeah, that's what I thought. Okay. Those weren't. I don't think I had different cell types.
Right here, I think this is.
Yeah.
So you see how if you go to the slide prior to that, you had those three: the red one, the orange, and the yellow, and they weren't doing much on the previous slide. And you say, and then so you look at the combination. You see the orange. You don't have it highlighted. It's just and it's like it's very weak, right? It's not doing anything.
Yeah.
But then when you go to the mitochondria, go to the next slide, the one you showed earlier.
Yeah.
Can you walk me through what I'm looking at? Now look at the macrophage. I'm sorry.
The macrophage, it's high there.
It's very high, but why is it caring about a macrophage but not anybody else?
We don't know. We don't know. I mean, and
that's my answer. But.
So this just mRNA is being processed, and there are different cell types are going to be turning over mRNA differently. There are pathways that will stabilize or not stabilize that are operating in different cells.
But couldn't you use that triple case just to look at macrophage by themselves?
No, exactly. Right. Our goal was to say, look, let's find one that works across. It may not be optimal for a particular cell, like a macrophage. I mean, if you really want to optimize, for instance, in a hepatocyte, you may just use a capped. But for the purpose of trying to generalize a system across many different cell types, we're going to build all these mRNAs with this backbone. That was our goal. Yeah. But because it's really hard. I mean, you want to, if all you do is look at hepatocytes, you might want to just optimize for the cap.
Thank you.
But that's a great question. Super good question.
Other questions? Oh, one more.
So at the beginning, you were talking about how the COVID-19 vaccine by Moderna, it used M1 pseudo-uridine, but you said that it had complications in it.
Yeah.
Could you elaborate more on that?
Yeah. So this guy right here, 5-methyluridine, is in the COVID vaccines. And yeah, it's pretty good, except that with time, that mRNA undergoes a frame shifting mutations. And so what that leads to is expression of a different protein other than spike. And they discovered that with these vaccines because what they were finding is that with time, people are expressing more than spike protein. And so you don't know what this other protein is going to be doing in your system. To avoid that complication, this guy doesn't have that problem. There's no reported frame shifting happening with this modification of the cytidine.
So are they talking about possibly changing that so they use the M5C instead of the pseudo-uridine?
You mean for the vaccines?
For the vaccines, yeah.
Yeah, well, I don't know
think
whether,
if you don't get modifications with the other one.
There's millions, billions of dollars that have already been put into that vaccine. So I'm not sure whether they're going to make that change.
A side question from, since pseudo-uridine causes these modifications, I recently read a paper in Dr. Valentine's class, actually. I'm probably going to give a presentation on this afterwards myself. It says that it's derived from biological responses. So, you know, I'm wondering why would it be present in biological systems if it is causing these modifications? Like, is it maybe not being seen as like
because
a sure
it blocks. What these modifications can do is that they're dampening the immune response. They're doing two things. One is the cap sequence is stabilizing the mRNA. As soon as that mRNA will go into the cytoplasm, the cell will chew it up unless it has that stable cap structure. And if you don't have these modifications, that mRNA will be recognized as alien and trigger an inflammatory response, basically a stress response in the cell. And so that's why, all of the mRNA therapeutic community has been going into this in detail. And we're basically building from that. We just basically went into that literature and said, okay, which is the best modification for us to be able to start introducing whatever protein we want in all of these different cell types in animals.
Right. Thank you. Other questions? If not, let's thank Jennifer again for a really wonderful
so
lecture.
much. Yeah.
Now the official part of the program is over, so the unofficial discussions and receptions, so you guys are welcome to explore.